Hello dear students and welcome to the second lecture. The topic for today will be quaternary structure and isoelectric point. Let's start with definition. What is this a quaternary structure? Some proteins contain two or more polypeptide chains that are called subunits and which may be identical or different. Actually, let's remind before that what was this tertiary structure. The tertiary structure, students, was one three-dimensional structure which is formed thanks to the information which comes from the primary structure. First, the primary structure was the sequence, type and number of amino acids that are connected between each other by peptide covalent bonds. After the formation of the corresponding alpha helices or beta sheets, which was called secondary structure, there is formation of one three-dimensional structure in which the main bond is disulfide bond plus different non-covalent bonds like hydrogen bonds, polar interactions, hydrophobic interactions, and if there is formation of more than one polypeptide chain, there will be quaternary structure. Let's try to draw something about the classical example of quaternary structure, which is hemoglobin. Hemoglobin, you can see, consists of one subunit, second subunit, third and fourth. All of them have their own primary, secondary and tertiary structure. And after the combination, thanks to non-covalent interactions, there is formation of one new protein with quaternary structure. If the bonds are covalent, it's not a quaternary, it will be tertiary structure. The most important, remember, the bonds between subunits are non-covalent. And especially the hemoglobin, which is the classical example, it is a tetramer or consists of 4 subunits or 4 polypeptide chains. Actually, students, in adults, the main hemoglobin, the major one, 95%, approximately 98% of adults, they have hemoglobin A1 is the name. Hemoglobin A1 means that consists of 2 alpha and 2 beta polypeptide chains. Let's see, where are they? 1 alpha, second alpha, 1 beta, and second beta. Do you know what there is inside the polypeptide chains? Yes? Yes. Yes, well done. It is a heme, students. In each polypeptide chain, there is one heme molecule, totally 4 heme molecules in one hemoglobin. In each heme, inside, there is one molecule of iron. How many plus, do you know? 2 plus, yes. The iron is 2 plus. If the iron turns to 3 plus, it won't be hemoglobin, but will be methemoglobin. This other structure with iron 3 plus will be called methemoglobin and cannot transport oxygen and carbon dioxide. Only this hemoglobin can transport oxygen and carbon dioxide. Other types of hemoglobin with different beta is hemoglobin A2 with 2 alpha and 2 delta polypeptide chains. In the fetus, there is different hemoglobin 2, which is called hemoglobin F, or fetal hemoglobin, with 2 alpha and 2 gamma polypeptide chains. This hemoglobin F has a higher affinity to oxygen. There are some other different more hemoglobins like hemoglobin E with epsilon chains and etc. You need to know that alpha chains are expressed continuously from embryo to adult. The difference chains are non-alpha that are changed during the development. Protein denaturation, the definition. This is the unfolding or disorganization of polypeptide chains. This is the disorganization of all structures without primary, which means disrupting of quaternary, tertiary, and secondary structure. And the denaturing agents are heat, organic solvent, strong acids or bases, different detergents, and heavy metal ions like elite. Sometimes there is reversibility of denaturation, which means if the denaturing agent is removed, the denaturation can be reversible, and proteins could remain again soft, again will be folding of the polypeptide chains, refolding of polypeptide chains after the removing of denaturing agents. The effect of solubility, denatured proteins are usually insoluble and may precipitate. Something about protein folding. Actually, students, everything in the polypeptide chain about the folding depends on the genetic information which comes from DNA, which means everything depends on the primary structure. After the information of primary structure, there is folding of secondary, tertiary, and quaternary. It's not obligatory all proteins to have quaternary structure. Maybe the last one can be the tertiary structure. And sometimes, if there is a different denaturing agent, the proteins can undergo misfolding and disruption of the polypeptide chains. For that reason, in our body, we have mechanisms to maintain the correct folding of polypeptide chains, the correct folding of proteins. Which are these mechanisms that assist protein folding? The first ones are called molecular chaperones, or just chaperones. The other name of these proteins is heat shock proteins. The heat shock proteins are used because of the facilitation of folding with correct interactions in the polypeptide chain to prevent aggregation. And chaperones use hydrolysis of ATP, or they use energy from ATP. One more action of chaperones there is, except this one, to protect correct folding of proteins. It is using of these chaperones in the signal transduction. Of steroid hormones. Why? Because, as you know from the last lecture, we talked about transport of compounds in the cytoplasm, in the blood. These places, you understood that these are water-soluble places, right? That's why the lipid-soluble compounds cannot be transported independently. In this situation, when we're talking about the steroid hormones, they need to be transported in the cytosol, and exactly chaperones facilitate this transport of steroid hormones in the cytosol. This is the second action of chaperones in the cell, except maintenance of the correct folding. They facilitate the transport of steroid hormones in the cytosol. The second mechanism or protein that help the correct folding these enzymes are called proline cis-trans isomerases. Actually, this bond between X amino acid, NE1, and proline is in trans configuration. Trans means that the one residue is like that, the second residue is like that. Cis configuration means in one direction that are located the radicals of amino acids. These changes can be catalyzed by these enzymes from trans to cis and opposite direction. The cis configuration is common in beta cells. And the third enzymes that are used to maintain the correct folding are called protein disulfide isomerases, PDI. These enzymes are used to change the places of disulfide bonds, because you understood that the disulfide bonds are the main bonds in the tertiary structure. They are used in the tertiary structure mainly, and that's why if there is incorrect formation of disulfide bonds, maybe will be incorrect folding. That's why we need changes of the places of these disulfide bonds. For this reason is used this enzyme, protein disulfide isomerases. For example, look here. Here we have one disulfide bond between, for example, amino acid number 1 and 2, and the second one between 3 and 4. But in the end, there is changes of the disulfide places between 1 and 3 and 2 and 4. This is the changes in the positions of the disulfide bonds. Why we need such of these mechanisms, especially the chaperones? Because students, many times in our individual life, we can have different situations with high temperature, different infections, diseases, and etc. Is it possible after all these situations to denature our proteins in our individual life? Of course not. That's why we need protection. But if the temperature is more than 40, 41 degrees Celsius, the protection cannot be very strictly. That's why until 40 degrees Celsius, the chaperones can be used for the correct folding of all our proteins, because we have, not only enzymes, all receptors, all many hormones, all transport proteins are proteins. Their nature is proteins or polypeptide chains that can be denatured. Here you can see some classes of chaperones that are called chaperones, that are used for the formation of protective cavities where unfolded or misfolded proteins can fold without aggregation, without aggregating. A relationship between protein structure and biological function. Here you will understand that if there is any changes in the protein folding, in the protein structure, no function. For example, there is misfolding of some enzyme. Where is the reason? In DNA maybe. And what will be the consequence? No catalytic function. Or maybe the misfolding or the defect is located in some receptor, but all receptors are proteins. What will be the consequence? No recognition with the ligands, no action of the receptors. And let's start with some altered protein conformation in some neurologic diseases. Actually, neurologic diseases can result when proteins adopt abnormal conformations leading to cytotoxicity or loss of normal function. The mechanism. Normal proteins may undergo misfolding due to genetic mutations, abnormal post-translational modification, or environmental stress. Misfolded proteins tend to aggregate, often forming better-pleated sheet structures, and the consequences will be with disruption of cellular homeostasis, impaired synaptic function, and trigger neuronal death. Examples are with Alzheimer disease and so-called prion diseases like TSE, which is transmissible spongiform encephalopathies. And let's start with the first disease, which is called prion disease. The prion disease in human is called also Creutzfeldt-Jakob disease, In animal, this disease is called bovine spongiform encephalopathy or mad cow disease. And what is the reason? The reason, students, is formation of one pathogenic prion protein which induces misfolding of normal prion protein without DNA or RNA involvement. And the mechanism is thus that this pathogenic prion protein acts as a template, and there will be accumulation of beta sheet-rich aggregates, and after that neurogeneration will be. The properties, these aggregates are resistant to proteases. Let's say something about this enzyme proteases. Students, the half-life or just the life of proteins in our organism is limited. For example, some proteins, I said about that in the last lecture, some proteins live maybe 30 minutes, some of them 5 minutes or 100 days, never mind. But they have some limited of life. After that, they need to be degraded. And there is absolutely strictly a regulation about the protein life. How many is? It depends from the DNA information for the formation of new ones, but not only. Not only there is a regulation about the synthesis, there is regulation about the destruction, about the degradation of old proteins. And these enzymes that are used for the degradation of old proteins are called proteases. Protein plus ase, proteases. These are enzymes that degrade old proteins. That's why if there is accumulation of some beta sheet-rich aggregates and no possibility of degradation, there will be accumulation between neurons and formation of this progressive fatal neurologic disorder. The last example is an Alzheimer disease. In the Alzheimer disease, students, instead of soluble alpha helix rich form, there is formation of beta sheet rich form, and which promotes self-aggregation. And in between neurons, can you see these brown plaques? These are the plaques which are called senile plaques, which means extracellular deposition of aggregated beta amyloids. And the second one, the second formation in the Alzheimer disease, there is formation of so-called neurofibrillar tangles with one hyperphosphorylated tau protein. This tau protein can be measured in the cerebrospinal fluid and the blood, and can be used for the detection of Alzheimer's disease. Can be used for the detection, this tau protein. Actually, why there is possibility of this self-aggregation of beta amyloid? Because there is impairment of the process of cleavage of one precursor protein, which is called amyloid precursor protein, APP. Other example of disruption of the protein and no functions are defects in the receptors. In this first figure, you can see one example of one disease which is called diabetes insipidus. Don't confuse diabetes insipidus with diabetes mellitus. Diabetes mellitus is this diabetes that you know, which is connected with glucose levels, insulin. It's not this diabetes. This diabetes, the reason is because of the impossibility of action of one hormone, which is called antidiuretic hormone. We talked about this hormone last time as one of the examples of oligopeptides. What was the other name of antidiuretic hormone? Do you know? It's a vasopressin. The other name is vasopressin. Here is written. And one more name, which is widespread, which is used more often, is arginine vasopressin. If you see this abbreviation AVP, this means vasopressin, or ADH, antidiuretic hormone, is the same. And what is the action? Because this hormone is a polypeptide, which means water soluble. This means that, actually it's a question, is it possible to cross the cell membrane? It's a polypeptide.
Yes.
I think more.
I think it's possible to cross.
No, students cannot cross the membrane because the cell membrane is a lipid soluble. That's why water soluble compound cannot cross the lipid soluble cell membrane, which means needs a receptor, membrane, a receptor to transmit the information. There is only transmission of information, but not crossing the membrane. That's why in this situation we have the cell. Here we have the corresponding receptor which recognizes the hormone. The receptors, this one is called V2 receptor of vasopressin. And what will be the consequence if there is some defect in this receptor? The consequence will be no recognition with the hormone, which means no signaling pathway. If the receptor is intact, is correct, with correct folding, the signaling pathway will be activation of one enzyme, which is called adenylate cyclase. After that, degradation of ATP to cyclic AMP, which is the second messenger of this hormone. Then cyclic AMP activates the protein kinase A, activates some other compounds and proteins in the cytoplasm, and in the end will be expressed on the membrane, but the outer surface of the membrane, some proteins, some transport proteins that activate the transport of water from the primary urine inside the cell and after that in the blood. This is the normal situation because the action of the antidiuretic hormone is antidiuresis, which means the resorption, opposite resorption of water from the urine to the blood, which means increasing of the blood because of the increasing of the water in our organism, and the result will be high blood pressure. But in this situation, we don't have intact receptor, which means no response of ADH, no response of vasopressin. This is the reason. We have intact hormone, but no receptor. The second example of defects in receptor is other disease, which is called familial hypercholesterolemia. Let's see one by one the parts of the words. Familial. What does it mean, familial?
Must be like genetic, so...
Must be genetic, must be in the family, must be inherited. This means inherited disease. Hyper, too much, high level. Anemia, most of you are from Greece, I think. What is this anemia? In the blood, of course. Anemia means in the blood. If the end of the word is urea, in the urine. Example: hyperglycemia, high glucose in the blood. Hypoglycemia, low glucose in the blood. Glucose-urea, glucose in the urine. Understand? With only one word, we can explain the content of something in some place. In this situation, we have this disease, but what is the reason of familial hypercholesterolemia? The reason of high cholesterol in the blood? The reason is a genetic defect. Most often, it's a mutation in LDL receptor gene. Let's see what is this. On the right of the figure, you can see, students, the normal situation. This is the membrane, the plasma membrane, and this is one lipoprotein. Lipoprotein, what is this lipoprotein? It's a combination between lipid and protein. Why? Because the lipids have a very big problem. They cannot be transported independently in the blood. They need transporters, which are proteins. That's why this combination is called lipoproteins. And one of them is called LDL. LDL-C means LDL cholesterol. Low density lipoproteins means LDL. People know that this LDL is so-called bad. But it's not bad the same, it's other one bad. But okay, for the beginning of your education in biochemistry, remember that this is the bad cholesterol. And the protein part of this LDL is called ApoB. The full name is ApoB100. This is the protein part of lipoprotein, which play a role of transporter, number one, and the ligand for the receptor of LDL. If there is intact receptor, can be possible recognition with the ligand, which is ApoB100. And let's see here, this is the LDL receptor, intact receptor. After the recognition with the ApoB100, there is formation of one receptor LDL complex. After that, there is internalization, which is called also receptor-mediated endocytosis, and formation of endosome. And in the endosome, there is releasing, thanks to the lysosome, the LDL particle and recycling of LDL receptors back to the membrane surface and can be used again and again. They're not destroying. They can be used again, the LDL receptors. This is the normal situation. One more. After the formation, after the releasing of this cholesterol, after that, it can be used for different processes like production of steroid hormones and etc., different derivatives from cholesterol. But let's see if what will be happen if there is defect of LDL receptors. Look, students, if there is LDL receptor mutation, is it possible the recognition with the ApoB of LDL? No, which means high level or hypercholesterolemia will be developed. High level of cholesterol because LDL cannot enter the cell, no recognition. And this is the first reason of familial hypercholesterolemia, and the reason is genetic defect in the receptor. But here are written some other mutations, not only in the LDL. I think it's very logical to understand that because the ApoB100, this one is a protein, can be mutated too, why not? It's a protein, which means the genetic information for the formation of ApoB100 comes from DNA too. That's why exists such a this mutation can also affect ApoB100, which is actually the ligand for LDL receptor. If there is mutation of ApoB100, no recognition again. It's another type of familial hypercholesterolemia. And there is one other type which affects the gene of this protein, which is PCSK9 gene. And the result will be increased degradation of LDL receptors. This is the action of this protein. If there is normal level of this protein, there is normal degradation of receptors. Normal. It can be regulated with normal amount of the receptors on the surface. But if there is gain of function, this type, gain of function mutation, there will be increased LDL receptor degradation, which means the same result, high LDL in the blood, which means atherosclerosis accelerating and premature cardiovascular diseases. Actually, students, this disease can be homozygote or heterozygote. In the homozygote disorder, the disease starts early in the childhood, and in the children there is formation of atherosclerotic plaques early in their life. Another example, no receptors, is expression of point mutation, and the example is very classical, sickle cell anemia. Where is the reason in this disease? can you see the normal hemoglobin on the right and the mutant hemoglobin on the left? Actually, with blue color, it's a DNA. With red color, it's mRNA. And in the end, this is the protein. The information comes from DNA. Actually, instead of thymine, there is adenine of the codon in DNA. And instead of adenine, there is uracil in the mutant hemoglobin. But this combination, guanine, uracil, adenine, leads to the formation of valine instead of glutamate. This replacement of hydrophobic valine instead of hydrophilic glutamate is a mutation which leads to polymerization and the precipitation of hemoglobin. And this mutant hemoglobin is called hemoglobin S, which leads to hemolysis of erythrocytes. Hemolysis means that the erythrocytes are broken down. This means hemolysis. The next example is a defect in post-translational modification of proteins. There are two examples, and this is the first one, which is about hemoglobin. Students, sometimes if there is a very high level of glucose in the blood, long-term hyperglycemia, you know this new term, long-term hyperglycemia, this glucose can be attached to hemoglobin. But let's discuss which hemoglobin, where is the place of hemoglobin actually. Students, tell me, where is the hemoglobin actually? Where is the place, the localization of hemoglobin, normally? Yes?
I think in the heart or the lungs.
Only?
They transport oxygen.
Absolutely, in erythrocytes. The correct answer is in erythrocytes. The correct answer is not in the blood, because many students say hemoglobin is located in the blood. Okay, but not alone. It is located in the erythrocytes, in the red blood cells. They are in the blood. And after their life, what is the length of their life? How many days they live? Approximately, the erythrocyte lifespan is approximately 120 days. 3-4 months. 2-3 months or 4 months. This is their lifespan, and after that they need to be degraded because they are called old erythrocytes. They have to be degraded from cells which are called macrophages, or cells from reticuloendothelial system. These macrophages can be in the spleen, in the liver, and different places. They degrade the old erythrocytes. And this is a normal degradation of old erythrocytes. But sometimes, some small percent of erythrocytes can be broken down inside the blood, not in the macrophages, inside the blood vessel. This is called intravasal hemolysis with some small percent. And this hemoglobin can goes out the erythrocytes and can be found directly in the blood, alone. Exactly this hemoglobin can be glycated non-enzymatically, which means spontaneously, without any enzymes. This is called glycation of hemoglobin. Students, imagine that you have sugar in your hands. You know how that the sugar sticks of your hands. Absolutely the same is when there is high level of glucose in the blood. It will stick to the hemoglobin directly. The question is when we have long-term hyperglycemia. It is normal. It is normal situation or not to have high glucose in the blood. No, student, it's not normal. Actually, after carbohydrate intake, the glucose in the blood is increased, but only 2 hours. This is, and after that, the glucose need to be decreased, need to be normalized to the normal range. But in some situations, it can be increased long term, and it is in untreatment, one disease. Which is this disease? With high level of glucose in the blood is this disease. Which is this disease? Yes? Yes, it's a diabetes. In diabetes, but non-treatment, untreatment diabetes, when there is no treatment with insulin, or new diabetes maybe, we have high level of glucose in the blood. And one more, don't forget that if you eat carbohydrate food with high glycemic index, will be the same. There is very high spike of glucose in the blood directly, and it can be happen not only in adults, in children too. Which means glycation, non-enzymatically glycation of hemoglobin can be happen in all people if they eat carbohydrate food with high glycemic index, or in untreated diabetes, mellitus, the usual diabetes. And after this combination between glucose plus hemoglobin, there is one intermediate formation of one sheet base between some residues of hemoglobin. There is in the end formation of this type of hemoglobin, which is called hemoglobin A1C. It is a glycated hemoglobin. This is an indicator for the blood glucose levels in the past 8 to 12 weeks or 2-3 months. Why? Because this is the lifespan of erythrocytes. And we can test how many is in the clinical laboratories, and the normal level is less than 5.7%. An example with defect in post-translational modification is scurvy. What is this scurvy? Is there a genetic defect? No, no genetic defect. Here the reason is another reason. The reason is a lack of vitamin C. No vitamin C. This is the cause. Dietary vitamin C deficiency. Why we need this vitamin? It's one of the examples of water-soluble vitamin, which is used, it's one of the reactions, which is used as a cofactor of 2 hydroxylation reactions. The first one is cofactor of prolyl hydroxylase enzyme, which hydroxylates proline to hydroxyproline. Hydroxylates lysine to hydroxylysine. These are 2 modifications that are used during the synthesis of collagen. Both of them are used in the formation of a stable collagen. If there is no vitamin C, no hydroxylation, and no formation of stable collagen, which means that can be, actually, where is the collagen? Do you know where is the location of collagen in the body? Yes?
In the skin.
Yes, in the skin, but actually in no connective tissue. In no connective tissue is abundance of collagen proteins. All blood vessel walls contain collagen. There is many collagen molecules around the blood vessel walls. That's why if there is no intact collagen, no stable collagen because of the vitamin C deficiency, there will be, the main symptoms will be bleeding. Gums bleeding, or poor wound healing, or bleeding from everywhere, from all organs will be. And In the end of the lecture will be some words about isoelectric point and protein precipitation. What is this isoelectric point or PI of proteins? Proteins contain amino acids with polar, charged or non-polar side chains. At physiological pH around 7.4, the charged residues contribute to the protein's behavior as a polyelectrolyte. The definition of PI is that isoelectric point is this pH at which the overall charge of protein is 0, or the number of positive and the number of negative charges are equal. This is the PI. At PI, the protein is electrically neutral, and that's why do not migrate in electrophoresis at this pH. What is the importance to know how many is the PI in proteins? Because exactly in this pH, the proteins can be discovered, can be studied about the purification, solubility, and electrophoretic separation, and they can be precipitated, and precipitated proteins can be denatured more easily. You can see this figure that in the acidic region, this is the pH, in the acidic region means here somewhere, the dissociation of acidic groups is suppressed and the net charge is positive, which means now we have protonated amino groups. And that's why the positive charges are predominate, they are present in this time, and as the pH increases, the net charge decreases, passes through 0, and then becomes increasingly negative. This is the alkaline region of pH. In this region, the dissociation of basic groups is suppressed. Basic group means the amino group. The dissociation of amino groups will be suppressed. And only the charge, that's why the charge will be negative, because of the presence of carboxyl negative charge at this time. For amino acids, that they have only two groups that can be dissociated. They are carboxyl group and amino group. The PI is the mean of the two pKa. This is the constants of dissociation of the amino acids. The amino in carboxyl group will be like that. For example, if we're talking about the glycine, for example, there are two dissociable groups, like amino group, that the pKa is around 9, and the carboxyl group, that the pKa is around 2. 9 plus 2, 11 is divided 2, around 5 will be the midpoint of both of the pKa, which is actually exactly this pH where the amino acid has isoelectric form. It is the isoelectric form with the equal number of positive and negative charge of this amino acid. Principle of electrophoresis. This is a key technique for the separation of proteins. It is used for the separation of proteins based on their charge and one more in the electric field. And we need two conditions to make electrophoresis. Number one is according to isoelectric point of the corresponding protein, and number two depends on molecular weight. They can be separated in gels, such as cellulose acetate, agarose, or polyacrylamide gel. And proteins migrate under the influence of electrical potential. The speed and the direction depend on their net charge and pH. Students, you have to know which are the serum proteins. that can be defined by electrophoresis. There are 5 types of serum proteins. Albumin, we talked about albumin last time. It was classical example of transport protein. Number 2, alpha-1 globulins. Number 2, alpha-2 globulins. Number 4, beta globulins, and 5, gamma globulins. And electrophoretic mobility depends on size, charge, and application. And here is the separation of serum proteins: albumin, alpha-1, alpha-2, beta, and gamma globulins. Here on the left, this one with red, pink, this one is an example of one electrophoregram. And this one on the right, this one, is called densitogram. The clinical use. Densitograms are used in diagnostics. If there is changes in the level of these serum proteins, we can put a diagnosis. For example, there is low peak of albumin, low level of albumin in the blood. What does it mean? To answer this question, we need to know which is the organ that produces albumin. This organ is a liver. If there is a liver disorder, some impairment of the liver, will be low production of albumin, which means this peak will be lower. And such this example, you can understand how you can put a diagnosis if you know this information. And not only albumin, there can be changes in all of these serum proteins to put correct diagnosis. And that's everything for today, students. Thank you for your presence today.